Hi, I'm Shahana Maji, and I'm from Dr. Greco's 9 a.m. Section NO4 Physics 2212 class. I'm here to talk about the public lecture that I went to called The Physics of Genes and the Promise of Personalized Medicine by Alexei Aksimentiev from the University of Illinois, Urbana-Champaign. I've always been really interested in how biology gets integrated into different kinds of sciences and engineering. I think this lecture really gets the point across that physics is necessary in all forms of science and can really be used to help make progress in biology and medicine. So, Aksimentiev first begins by defining DNA. It's kind of a blueprint for genetic information, and that genetic information is used to create different organisms. Chromosomes are structures that hold this DNA, and they're responsible for why people are different individuals. We look different, sound different, things like that. The Human Genome Project was a project that began in 1990 and ended in 2003. It's where we realized that humans aren't actually as different as we thought they were. They, in fact, share about 99.4% of of the same DNA sequencing. And for a little context, humans and chimpanzees actually share about 99% of that same DNA sequencing. DNA is comprised of nitrogenous bases. Those are the A, C, T, and G things. A sugar called deoxyribose and a phosphate group. This phosphate group is negatively charged, which thus makes the whole DNA strand or DNA polymer negatively charged. This is where some physics comes in. The negatively charged DNA molecule gets attracted to positively charged ions, especially when in solution. So these positively charged ions can end up serving as a bridge between two different DNA molecules, like so. Here you go. As you can see, the negative charges represent the DNA polymers, and the +2 cations, which are the multivalent ions, end up serving as a bridge. So the negatively charged ions get attracted and start moving towards that positive 2 charge. All right. DNA gets turned into RNA, which then gets turned into proteins, and proteins are vital for cells and the body in general. So all cells actually contain the same exact DNA, but what differentiates those cells is what genes are turned on and off within the DNA. So the turning on and off of these genes is called gene expression, and epigenetics works with modifying gene expression as opposed to modifying the genetic code itself. Over time, scientists have discovered that epigenetics occurs at the histones, and histones are it's like a core of proteins surrounded by coils of DNA, and that happens through methylated lysine. These three components together comprise a nucleosome, and I drew a nucleosome here. So, yeah, this nucleosome obviously is not drawn to proportion or anything, but this is the histone core, which is again proteins, and then the methylated lysine allows the DNA coils to surround the histones. And yeah. It turns out there's some really cool physics involved with nucleosomes. There's actually something called the Blue Waters supercomputer that Aksimentiev alluded to, or I guess taught us about. Basically, it computes forces between nucleosomes, and you can obviously see some physics being used there. Physics is also involved in nucleotides because GC-rich sequences of DNA are actually less attractive than AT sequences of DNA, and that becomes important later with DNA sequencing and electrophoresis. Methylating DNA additionally increases its attractiveness. Its attraction. Yeah, it increases DNA's attraction. Um, Sequencing the human genome has definitely decreased in cost over the years, thanks to improved technology. In the lecture, we learned about many different kinds of methods of DNA sequencing. One is non-pore electrophoresis. Basically, DNA gets put into a chamber that is positively charged on one end and negatively charged on the other. Since the DNA is negatively charged, it will start moving towards the positive end, and ionic current then, I guess, is measured through these pores, and the blockade of ionic current reveals the sequence of the DNA. Um, this is kind of what they do in crime shows to like match people's DNA or look for like paternity testing and stuff like that. We in the lecture we also learned about Minion, which was created by the Oxford non-pore technologies. It reads 100,000 nucleotides through 800 parallel detection wells with 80 to 90% accuracy. And then we also learned another method of DNA sequencing called quantum sequencing, and that uses electrode tunnels for each nucleotide. I think the coolest part of this lecture was the like what next portion of the lecture. Basically, there's two main goals in medicine right now that he talked about, and one of them is targeting. Actually, sorry, I should probably go back and say that cancer is a direct disease of the cells and the DNA itself. So two major goals for cancer prevention or cancer treatment is to one, target the cancer cells directly instead of hurting, like in chemotherapy. Chemotherapy actually destroys healthy cells in addition to cancer cells, which is terrible. So they're trying to minimize that. And the second goal is to actually prevent the degradation of drugs. Like when drugs are getting put into the body, um, they decrease in their usefulness because of, I guess, different processes in the body. So one way to combat that is the idea of DNA origami, and basically the idea is that you can structure DNA in a certain way that it kind of becomes like a box to hold drugs in, and then that gets put into the body, and then antibodies can locate and get into cancer cells, open that DNA box, and release the drugs directly into those cells. So I think that was an amazing idea, completely mind blowing. I think it was really cool. Throughout the whole lecture, I was really enthralled. My eyes were kind of opened up as to how physics really needs to work. I guess biology, in order for biology to work, it really needs a lot of physics and vice versa. So it was really cool to me that DNA can you can actually get kind of creative with DNA with this DNA origami idea, and DNA can potentially save thousands of lives in addition to creating it. So, thank you.